Allosterics. And this is our discussion of sort of the non-competitive inhibition. Okay? The definition of allosterics, like I said, allos in Greek is just other. So in sort of a really general sense, it is communication between two sites on a single protein. Okay? That's important. Two different proteins, one binding to something and then it affecting a second protein is not allostery. Okay? We're talking about two groups on a single protein. Often on two different subunits, that's totally fine. But we're talking about, one protein could be an oligomer, different subunits. Okay? So there's the active site and then the site of modification. Allostery provides an important regulatory mechanism in a lot of cellular and metabolic pathways. This is how a lot of things work. Okay? I think the only example we really talk about in detail is hemoglobin. Okay? A lot of things function this way. Allosteric proteins are critical for sort of the responsiveness of cells, okay, to external signals. So here we have two different examples of allostery in action. It could either be inhibitory, or allosterics could be activating the protein. So in the top, we have two different sites. The active site, we have our enzyme. We have our secondary site, our allosteric site. When the allosteric inhibitor binds, okay, which is, like I said, you could design drugs that function either way. Either if you want to turn something on because it's not functioning properly in your body, or if you want to turn something off because it's like running continuously. Allosteric inhibitor binds. It changes, there's sort of poor drawings, but it changes the shape of the active site so that the substrate can't bind there anymore, or if it was bound, it'll kick it off, whatever it was. Alternatively, this one's a little bit more obvious of a shape change. I didn't draw these. When the allosteric activator binds, it changes the active site from sort of some distorted or shape where the substrate that the enzyme normally wants to bind, its regular ligand can't bind, changes to the shape such that it can, and you can turn it on. Okay? So allostery could work either direction. In order to get biological function, proteins and other molecules have to be able to switch between these sort of on and off states. The switching events that alter protein activity typically come from binding and often allosteric binding. Okay? So generally, when something binds, it either turns it off, or when something binds, it either turns it on. Okay? And that's how a lot of pathways in the body will function. Okay? So we can think of sort of this one-to-one binding, this one input produces one output. Your book calls it information processing. Use this if you like. The more important word that's maybe you might see in some of my questions and it'll be important to maybe understand is we'll call this like a graded or linear response. So substrate binds to enzyme, that's sort of your input. Enzyme converts it to a product, output. Bind, turn into output. Same thing here, sort of maybe a hormone binds to a receptor in the cell membrane, and then something on the interior gets phosphorylated. That's one input, one output. Like binding is the input, and then whatever the output is. And again, like I said, once something's phosphorylated, there might be downstream internal things. You release more calcium, you make the nucleus do something, who knows. Okay? Same thing here, a transcription factor binds to DNA, transcribes the gene. Like one-to-one input output. We call this linear response or graded response. Okay? So an output function that depends on the input in this hyperbolic fashion where the output switches from about 10% to 90% over that 100-fold change in input strength, that universal binding curve, is what we call, again, this graded or linear response. We said the hyperbolic shape was our quintessential. We know we have sort of a one-to-one situation going on. Okay. Can you think of any issues with the one-to-one case where switching from 10 to 90% happens over a 100-fold change in the concentration of your input, or the concentration of your hormone, the concentration of your substrate, the concentration of your transcription factor? Any thoughts about real life why this probably doesn't work? Do you think 100-fold concentration difference are practical in the body? No. So this would be, basically, it's imperceptible to small changes. Small changes would do nothing. 100-fold changes aren't really realistic. This also is really slow. So this sort of process of one-to-one would be way too slow for sort of a practical application. Okay? Interactions would either be too slow or require too large a fold change in ligand concentration to be biologically useful. Okay? But that's really useful for drug design, right? Because we can deliver something at 100-fold concentration and, interrupt what's going on if we want to very easily. But biologically speaking, the normal concentrations and physiological concentrations of things aren't going to have these big swings. Okay? So rather what we'll have is we have to have ultrasensitivity. An ultrasensitive system is one that can switch from on to off over a much smaller range. Okay? Something smaller than 100-fold. So in our binding example, fractional saturation was going from 10 to 90 roughly. Over 100-fold concentration change in an ultrasensitive system, it will be much smaller. Okay? So here's what it looks like on the universal binding curve. Okay? So it's narrowed to maybe even a 10-fold change or possibly even smaller, depending on how ultrasensitive the system is. Another important thing to notice is, again, our typical one-to-one input to output binding is typified by what we call linear response. We call it graded response. We have a hyperbolic isotherm. Okay? For an ultrasensitive system with this faster or more rapid switch from on to off or bound to unbound, it's going to be typified by a sigmoid or an S-shaped curve. Okay? So again, if you see a graph and you're asked, is that one-to-one binding and it's a sigmoid? It's really easy to just go, nope, the curve wouldn't look like that. Definitely has to be some sort of other thing going on. Okay? So like I said, just note some of the hallmark shapes of the different curves. That's what we call an ultrasensitive response, one that's sort of flipping really rapidly in a smaller concentration range.